Okay, so as you all know, my name is Lydia. I am currently co-advised by Professor Cunningham and Professor Harley on this project called Photonic Crystal Enhanced Microscopy for Studying the Invasiveness of Glioblastoma Multiforme Cells. So those are a lot of long words, so I'm going to go into detail on what everything means. So first I'm going to go over the background of what I'm studying, the glioblastoma cells, and what the normal brain looks like, and what PCEM is, and how we're using it to study GBM invasiveness. So the normal brain is composed of neurons and helper cells called glial cells. And of these glial cells, there are oligodendrocytes, microglia, and astrocytes. And of these, I'm specifically looking at astrocytes. And in addition to the cells, there's also the ECM composition. So as opposed to most other parts of your body where you have a lot of collagen and fibrous proteins, in the brain you have a lot of you have a lot of hyaluronic acid, which is sort of like a glycoprotein, and also you have low fibrous protein content, so less collagen and fibronectin that you would find in other places in your body. So in particular, I'm looking at glioblastoma multiforme, which is one of the most common and aggressive and invasive brain cancers. The median survival for people with glioblastoma is 14.6 months with chemo and radiotherapy and 4 months without any treatment. And the 2-year survival rate is 30%. And this is just an MRI image of someone with glioblastoma, and you can see that they have this tumor mass here. So glioblastoma is a grade 4 glioma, and that's the worst stage of glioma. And as I said, we're looking at astrocytes, so this is called an astrocytoma. And this is a highly vascularized tumor mass, and the cells are extremely proliferative and invasive or migratory. So there's very diffuse invasion, meaning so they don't usually go out and metastasize to other organs. Mostly they think it's because of the blood-brain barrier, so it prevents things from going out. And it's also highly specialized ECM, as I said earlier. Instead of like the fibrous proteins that you find elsewhere, there's a lot of hyaluronic acid. So these factors play a role in preventing metastasis of the cells, but sort of increasing the diffuse invasion. And because the cells are so diffusely invading the brain cell, this is why it's very difficult to treat. And also GBM cells constitutively produce HA, which is the hyaluronic acid that I talked about before. And as opposed to normal cells that are found in the brain, there's an increased production of fibrous proteins by GBM cells. Some of the challenges to GBM therapy include the blood-brain barrier because it's selectively permeable, and also the diffuse invasion that I talked about earlier. So these are two different patients, and so GBM cells have a tendency to move more on the white matter tracts in the brain. And so the corpus callosum is one of the largest white matter areas in your brain. And so you can see that there's this huge tumor here. And also there's this diffuse invasion, diffuse spreading of the tumor cells throughout the brain. Also, there is a problem of cancer heterogeneity. So there was a recent paper that looked at the heterogeneity of a patient's brain tumor, and they were able to they had 4 different clones, and these 4 different clones had 4 different phenotypes. And so this is also one of the problems in treating GBM. So what I'm looking at specifically is I'm trying to understand the interaction of the ECM with the GBM cells that cause it to be so diffusely invasive. And this is in hopes that it'll lead to better diagnostic and treatment methods. So now I'm going to go into what photonic crystals are and how I'm using them to look at GBM invasiveness. So PCEM is the method that I'm using. It stands for Photonic Crystal Enhanced Microscopy. So photonic crystals, the ones that we are using, have a a periodic grading structure. So this can be in like one-dimensional, two-dimensional. The ones that we are using are one-dimensional gradings, and this is one requirement is that it needs to have a dielectric material. So the dielectric material that we're using here is TiO2. And yeah, so what happens when you have this grading at this nano scale, you end up having these areas that are like, this is the power distribution, and you also have this evanescent field. And what I am looking at is when you have something that is going into this evanescent field, there will be a shift in the wavelength. And so I'm looking at this wavelength shift in response to a cell getting on top of the substrate and then adhering to it. So this is the PCEM instrument that we use. This is the microscope, and there's, I guess there's the stage, and this is this is sort of like an incubator where it's maintained at 37 degrees and 5% CO2. It's not exactly sterile, which has been a problem, as you will see later when I show you my data. But yes, so the hypothesis is that this wavelength shift corresponds to a change in the refractive index, which we hypothesize is because of the cell getting onto the substrate and starting to engage with the substrate, which will be coated with some sort of matrix component, such as hyaluronic acid. And later I would like to look into what proteins or factors play into this, mainly integrins and focal adhesion complexes. So the question that I'm asking is, can I correlate the wavelength shift with GBM adhesion, spreading, and motility? So the preliminary experiment that I ran when I was doing the rotation was that I compared GBM adhesion and motility on uncoated PC surface and a coated PC surface that was completely coated with HA. So this was two separate PCs that were run on two separate days because you can't run them simultaneously. So these are the results that I got. So basically when there is nothing, I guess there will be a resonance at 621 nanometers of like that's the wavelength. But as cells start to adhere, you see a redshift in the wavelength. So it's like, oh, what are all these colors? So that was one of my questions, and what we're trying to do right now is trying to see how do these color changes correspond to how the cell is adhering. And this is a video comparing with and without HA substrate on the substrate, and yes. But this is very difficult to understand what's going on. So when you compare with the bright field image and the PCEM image, you can sort of correlate where cells are. But one of the problems was that since we ran it in two different PCs on 2 different days, and this was one of the first experiments that I ran, like it wasn't very controlled, and one of the problems was that it was difficult to quantify motility. That's something that we're still working on, but one of the difficult things was that there was an inconsistent cell count. So if you remember, there were a lot more cells for without HA, which was the first experiment that I ran, as compared to the later experiment. And the problem with coating the entire PC surface was that I wasn't able to measure how thick the HA was, because there was everything was coated with HA, and so I couldn't compare how it compared with just a bare PC unless I compared it to another separate PC. So what I decided to do after this was then I would print or spot an HA spot on a PC and compare how the cells are moving at that interface. So these are the results. So you can kind of, Okay, so you can kind of see here that there's this interface, and you can see it better here. Because the HA is now on top of the PC, you see redshift also where the HA is. So that was good. But one of the problems was that after 3 hours, all the cells died. And yes, and then, yes, there were, yes, cells died. And we thought maybe something went wrong, and we thought it might be because of the cell substrate. So, not the PC substrate. So the PC substrate is not supposed to be UV, is not supposed to be exposed to UV. But before I realized this, I thought I would sterilize it with UV for just a little bit to keep it sterile so that cells don't get contaminated. But apparently that was bad, and the PC, even for 10 minutes, it was ruined. And so that's why we can't, you can kind of see that these dots don't really correspond to the cells, and we think that that's because these are not actually cells, but they're just problems that occurred because of the UV exposure to the PC. And also, so the cells are supposed to start spreading within an hour, yes. But this didn't happen. And so in the next one, I said, well, I won't use UV, but I'll try to keep everything as sterile as possible. So using these as sterile as possible methods, I got this image where this time, instead of having this huge interface, huge HA interface, I wanted to see maybe if I had a spot, I can see how the cells are moving on it and how they migrate towards this HA spot. So this is what I was trying to see. And I was able to see this for the first hour. And you can kind of see, so here, these do correspond with the cells. One of the problems, though, was that I wasn't able to see any of the cells that were actually on the HA. And this time, it was because the HA was too thick. And so, at least this is, yeah. So if you remember from the earlier image that I showed with the evanescent field, the evanescent field goes up to 400 nanometers. And so that is the region where you can see this wavelength shift. But there was no wavelength shift at all on the HA. And this may be because the HA was too thick and it went past the 400. And so it's just, there's no wavelength shift that is visible. So this is another thing that I'm trying to work on. But also there was another problem of contamination again. You can kind of see, in the background, you start seeing all these little dots. And this time this was actually bacterial contamination. And so we realized, well, I realized with the help of the postdoc who was helping me, that so we have a stage, and the stage, so as I mentioned earlier, the incubator, or I don't know if I mentioned earlier, but the incubator that I'm using with the stage inside is not completely sterile. There's no UV, and it's not complete, it's sealed off, but everything has to be cleaned with ethanol, which I did. But every time you want to take a bright field image, you sort of need to, open it and so the PC you don't want the PC to be under the light, so we cover it. But then in order to take the bright field image, there is no built-in light to the microscope. And so what we do is use the ambient light. And in order to do that, we need to open it and allow the ambient light to get in. And so when this happens, there's a lot of potential for contamination. And since there's no blower or no UV, it doesn't prevent things from going in. So previously in the first experiment, I ran it for 6 hours and there was no contamination with that. And that was because we had a holder that sort of prevents outside air from getting in. But this time it was misplaced. Well, it is now found, so I can use this for my next experiment. So the sterile environment problem is sort of solved, but this is one of the things that I'll be doing again for studying the GBM attachment and motility for at least 6 hours. And yes, And so what I want to do in the future after getting the PCEM data is to also look at what are the exact proteins that are playing a role in GBM attachment and motility. So some of the proteins that are produced by GBM cells that are known to be produced by GBM cells is collagen, fibronectin, and laminin. So these are some of the proteins that I would like to look at. And also there are integrins and focal adhesion complexes that are formed in cells when they adhere to a surface and start moving. And so these are also some factors and proteins that I would like to look at. So I'll be using methods such as Western blot or immunofluorescence in order to detect these proteins. Yes, so that is all that I have to share today. But yes, so I would like to acknowledge my co-advisors, Professor Brian Cunningham and Professor Brendan Harley, and the postdocs, I guess they're all postdocs now, who are helping me on my project. So Dr. Uezo, she has been helping me a lot with the PCEM equipment, and Dr. Sunny Choi and Sarah Padron, they have been helping me with the, Sunny has been helping me with, she's the one who helped me initially with the culturing and coming up with the biological problem that we're looking at. And Sarah is the one who actually works with GBM cells, and so she's the one who has given me that part of that side of the knowledge. Yes, and I would also like to thank the IGERT traineeship, which allowed this project to be possible. Thank you. Do you have any questions? Yes.